You know, if you really stop and think about it, whenever we talk about getting a medical diagnosis, there is this underlying, almost comforting expectation of absolute precision.
Oh, absolutely. We treat it like mechanical engineering.
Exactly. Like if you fall off your bike and your arm swells up, you go to the hospital, they take an x-ray, and there it is. That stark, jagged white line across the radius bone.
It's binary. I
Uh-huh.
mean, it's either a fracture or it's a sprain. There's this deep psychological comfort in being able to put a name, and a clean physical boundary
Yeah.
on what's hurting you.
Right. We desperately crave that kind of visibility. We like our biological problems cleanly categorized in these separate little boxes.
We really do.
But then you step away from the emergency room and into the world of viral pathogenesis and national data surveillance, and suddenly that crisp, high-definition X-ray machine is just, it's shattered. It's completely broken.
Yeah, you're not looking at a clean picture anymore.
No, we are looking at a diagnostic landscape that is honestly incredibly murky.
It is the absolute definition of diagnostic muddy waters. Because when you start dealing with complex immune responses in natural databases, you leave the realm of binary fractures and you enter the realm of statistical noise and, well, microscopic camouflage.
Because what if I told you that a pathogen could pull off a microscopic magic trick? A biological mechanism so unimaginably sophisticated that it doesn't just attack your cells, it actively turns your body's own defense systems, your own internal communication networks, against itself.
Which is terrifying on its own.
Right, but then what if I told you that when the fallout from that specific magic trick finally hit the general population, the institutional blind spots of the United States top safety algorithms fundamentally failed to see it happening?
It's a terrifying paradox to confront. I mean,
Yeah.
we live in an era where we always assume that gathering more data automatically provides more clarity.
Yes,
We
exactly.
think, if we just have enough data points, the truth will obviously reveal itself. But in this specific case, the sheer unprecedented volume of information actually blinded the experts to the danger.
The noise swallowed the signal.
The noise entirely swallowed the signal.
Which brings us to the mission of today's deep dive. We're going to completely decode the molecular blueprints of SARS-CoV-2. We want to trace exactly how this biological machine is built down to the atomic level, and then we are going to follow the data trail to see how an unprecedented flood of pandemic reporting physically broke the government's mathematical safety nets.
And to do this, we have gathered a massive, incredibly dense stack of sources for you today.
It is a heavy stack, let me tell you.
It's a mountain. We are pulling from advanced structural physics analyses, deep bioinformatics papers, declassified DARPA grant proposals, and a fascinating, highly contentious trove of internal FDA email threads that were actually subpoenaed by a Senate subcommittee recently.
And because we are pulling from congressional subpoenas and virology debates, we need to explicitly state the ground rules for how we are handling this material today.
This is crucial for anyone listening. We are leaving absolutely all politics at the door.
Every single ounce of it.
We are not taking sides in the lab leak versus natural origin debate. Our goal is not to litigate that at all, nor are we going to endorse any specific political framing found within the rhetoric of these Senate reports.
Right. Our job today is to act purely as neutral forensic data accountants.
Exactly. We are looking strictly at the raw genetic code, the underlying mathematical models, and the literal timestamps on institutional communications to understand the mechanics.
We want to understand the mechanics of the pathogen and the mechanics of the bureaucracy.
Just the data. We want to understand how the machine was built, how it operates under the extreme stress of the human immune system, and what actually happened when the architects of our public health surveillance machine raised their hands and said, hey, the math isn't working anymore.
Okay, so let's unpack this, because before we can even begin to understand how a virus broke a government safety algorithm in 2021, we have to understand how the virus itself was built and exactly how it breaks into a human cell. And honestly, if you're like me, your common assumption is that a coronavirus is just this passive, tiny, spiky ball of infection just floating around in the breeze waiting to bump into something.
Yeah, that is the popular image we see on the news, right? The gray ball with the red spikes.
Exactly.
But looking at the genomic architecture in these papers, that framework is totally insufficient. It vastly underestimates what we're dealing with. The SARS-CoV-2 genome is a positive sense RNA strand of about 30 kilobases in length.
And just to put that in perspective for you, for an RNA virus, 30 kilobases is absolutely gargantuan.
It's massive.
Why is that so unusually large?
Because RNA itself is highly unstable. It degrades rapidly in nature. Most RNA viruses, like the common cold, are incredibly small because if they get too long, the genetic code literally snaps under its own weight or it accumulates too many fatal typos during replication.
Oh, wow. So they just fall apart.
Yeah, they fall apart. But coronaviruses push the absolute structural limits of how much code you can pack into a single strand. They manage to hold 30 kilobases together because they encode their own complex proofreading machinery like an internal spell checker to fix genetic typos as they replicate.
Which is fascinating, but I want to circle back to a phrase you just used, positive sense RNA. The sources hammer on this, and the best analogy I've found to explain positive sense RNA is to think of it like a pre-programmed, auto-executing USB flash drive.
That's a really great way to visualize it.
Right. If a virus has what they call negative sense RNA, the code is basically written backward. The virus has to enter your cell, unpack its bags, and spend crucial time building a translator before it can actually do anything. It's a delay. But positive sense RNA is perfectly formatted for the host.
It's plug and play.
Exactly. The absolute millisecond that viral RNA hits the cytoplasm of your cell, your own ribosomes, which are basically your cellular 3D printers, read that viral code as if it were native-friendly instructions.
They just start printing viral proteins immediately. There is zero delay. It is an instant, highly precise hijacking. And traditionally, people tend to focus on the structural proteins, the spike, the envelope, the membrane.
The outer shell stuff.
Right. But treating the virus as just a passive delivery vehicle entirely misses the actual mechanism of the disease. To truly grasp the pathology, we have to stop thinking of it as a spiky ball and start viewing it through the framework of an active saboteur.
An active saboteur. This blew my mind when I was reading the structural analyses. We were talking about the non-structural proteins, or NSPs.
Yes, the NSPs.
When the virus enters your cell, its very first act isn't just to blindly replicate. It deploys a biochemical hit squad. I want to look specifically at the documentation on NSP1. The texts literally refer to it as the cellular saboteur. NSP1 is the tip of the spear. What is its exact mission?
Its primary job is to execute a scorched earth campaign against your cell's internal communication networks.
Okay, let's break that down.
Let's look at how a cell normally defends itself. When a human cell detects a biological breach, its immediate response is to manufacture proteins called interferons.
Interferons.
Right. Think of interferons as distress flares. The infected cell shoots them out to warn all the neighboring cells to pull up their drawbridges and lock their doors.
Okay, so the cell senses danger and it needs to fire a flare.
Yes. And to fire those flares, the cell's nucleus, the command center, sends messenger RNA, which are the blueprints for the flare, out to the ribosomes, the 3D printers we talked about.
Right, the printers need the blueprints.
But this is where NSP1 comes in. NSP1 physically wedges itself right into the entry channel of your ribosomes. It acts exactly like a cork in a bottle.
So it physically blocks the cell from reading its own distress flare blueprints.
Exactly. It plugs the machine.
Yeah.
But it goes a step further than just blocking. NSP1 then activates an enzyme that literally shreds your cell's messenger RNA into useless pieces. It cuts the phone lines.
But wait, I'm reading this and thinking, if the virus is corking the ribosome and shredding the cellular blueprints, how on earth does the virus print its own proteins?
Right, that's the paradox.
Doesn't it need those exact same cellular 3D printers to build the viral army? How does it survive its own scorched earth tactic?
This is where you see the sheer paranoid genius of the sabotage. The viral RNA has a highly specific leader sequence attached to the very front of its code.
A leader sequence?
Yeah, it acts exactly like a VIP pass at a club.
Yeah.
NSP1, the cork, is programmed to recognize this specific VIP sequence.
Oh, wow.
So when human mRNA tries to enter the ribosome to build a distress flare, the cork stays tightly in place and the human mRNA gets shredded. But when the viral mRNA approaches, NSP1 temporarily steps aside, lets the viral code slide through the channel, and then slams the door shut again.
That is unbelievably sinister. It's monopolizing the entire manufacturing capability of the cell while simultaneously silencing the distress signal.
It's total control.
And the coordination, the sheer paranoia of this hit squad doesn't stop there. The genomic data highlights another protein called NSP12.
Yes, NSP12 is crucial.
Now, NSP12 is the RNA-dependent RNA polymerase. Basically, it's the photocopier that replicates the virus's genetic code. But the photocopier has a weapon attached to it, right?
It does. The structural data reveals that NSP12 has a completely distinct secondary function outside of just copying. It actively targets a crucial transcription factor in your cells called IRF3.
Okay, what is IRF3?
Think of IRF3 as the security guard who holds the keys to the cellular armory. When the cell detects an infection, IRF3 is supposed to rush into the nucleus, the main command center, and turn on hundreds of antiviral defense genes.
So it's trying to unlock the weapon.
Exactly. But NSP12 physically intercepts IRF3. It tackles the security guard in the hallway before he can even unlock the weapons.
So it's copying the virus while actively holding the door to the armory shut. This is like a heist movie happening at a microscopic level.
It really is.
And then we have the accessory proteins, like ORF6. The data shows ORF6 goes after the nuclear pore complex.
Yes, the nuclear pore is the actual physical gateway into the nucleus.
Yep.
And it is heavily guarded by a protein called NUP98. NUP98 acts like a bouncer at an exclusive club, strictly regulating which signaling molecules are allowed in and out.
Okay.
ORF6 physically binds to NUP98 and hijacks it. It essentially bribes the bouncer. By doing this, it completely blocks the nuclear import of two vital signaling molecules called STAT1 and STAT2.
And STAT1 and STAT2 are the molecules that respond to those interferon distress flares we talked about earlier.
Exactly.
So ORF6 ensures that even if a neighboring cell somehow sees a flare and gets the warning, the message cannot be delivered to its own command center to mount a defense. The entire signaling pathway is paralyzed.
It's a complete blackout.
The level of coordination here, corking the ribosomes, intercepting the security guards, bribing the bouncers, is staggering. But to execute this internal, coordinated sabotage, the virus first has to breach the exterior wall of the human cell. And that brings us to the thermodynamics of binding.
And this requires us to zoom in microscopically on the S1 domain of the spike protein.
Right.
Within S1, there is a very specific sequence of amino acids called the receptor-binding motif, or RBM. This is the molecular fingerprint. It acts like a highly specialized grappling hook that dictates exactly which species a virus can infect.
And SARS-CoV-2 uses its RBM grappling hook to target the human ACE2 receptor. And the binding affinity, the sheer grip strength, is practically off the charts.
It is.
How far off the charts are we talking?
Well, the structural physics analyses show that the SARS-CoV-2 spike protein binds to the human ACE2 receptor approximately 1,000 times more tightly than the spike protein of its closest known bat relative, a virus called RADG13.
1,000 times. That isn't just a minor evolutionary tweak. That is an exponential leap in biological efficiency. How is that physically possible at the atomic level? How do you get 1,000 times more grip?
It comes down to structural physics and the actual shapes of the molecules involved. If we look at the sequence alignment, we see microscopic swaps in the amino acids right at the contact interface. Let's look at position 486 on the spike protein.
Okay, position 486.
A bat virus, RADG13, the amino acid at position 486 is leucine. Leucine is an aliphatic amino acid. In plain English, it's relatively small, basically a short straight chain of carbon and hydrogen atoms.
So a small straight chain.
But in SARS-CoV-2, that small leucine has been replaced by phenylene.
And phenylene is massive by comparison. It has a large, bulky, aromatic ring of carbons.
Exactly. It's a bulky structure.
Yeah.
Now, on the surface of the human ACE2 receptor, there happens to be a very specific deep hydrophobic pocket. Hydrophobic meaning it repels water.
Right.
In the chaotic, watery environment of the human bloodstream, hydrophobic molecules desperately want to clump together to escape the water. The bulky phenylene ring on SARS-CoV-2 perfectly inserts itself into that hydrophobic pocket on human ACE2.
So it fits right in?
It drives deep into the crack and locks it into place with immense stability because both sides are trying to escape the water. The bat virus simply doesn't have the bulk or the reach to get into that pocket.
Okay, I love this visualization. So it's the difference between pushing a flat hand against a smooth wall versus driving a metal piton deep into a crack in a cliff face and hooking a carabiner to it.
That's a perfect analogy.
One slips off, the other can hold your entire body weight, but the physical bulk is just one part of the grip. The sources also mention an electrical charge element, right, at position 493.
Yes, the electrical component is fascinating. At position 493, SARS-CoV-2 has an amino acid called glutamine. This glutamine forms what structural biologists call a salt bridge with the human ACE2 receptor.
A salt bridge.
A salt bridge is a combination of hydrogen bonding and electrostatic attraction. It occurs when a positively charged molecule and a negatively charged molecule get close enough to essentially snap together like high-powered magnets.
Wow.
And the bat virus lacks the specific amino acid required to generate that magnetic snap.
So by swapping a short carbon chain for a bulky ring and adding a molecular magnet, the virus becomes 1,000 times more effective at breaking down our cellular doors.
Exactly.
Which naturally forces anyone looking at this data to ask, how do scientists even trace where these highly specific microscopic changes came from. Which brings us to the evolutionary footprint debate and the incredibly complex mathematical models virologists use to read the history of RNA.
And this is where the math gets incredibly dense.
Mm.
The sources outline a whole suite of algorithms used to build phylogenetic trees.
Right.
A phylogenetic tree is essentially a viral family tree, attempting to map out who begat whom over thousands of years. Broadly speaking, these algorithms fall into distance-based methods, like neighbor joining, and character-based methods, like maximum likelihood.
Let's break those down. What is neighbor joining?
Neighbor joining is the older, simpler method. It basically measures the raw genetic distance between every single pair of viral sequences in your dataset. It literally counts the number of differences, the typos between two genomes, and it pairs up the closest neighbors first.
So it just clumps the most similar ones together.
Right. It builds the branches backward to a common trunk based purely on who has the fewest differences.
But the bioinformatics papers point out a massive limitation there. Basic distance methods assume all genetic mutations are created equal. They assume changing an adenine to a thymine is just as likely to happen in nature as changing an adenine to a guanine. And biology absolutely does not work on perfect, equal coin flip probabilities, does it?
Far from it. Chemistry dictates probability. And that is why evolutionary biologists developed nucleotide substitution models. The texts detail how this math progressed over the decades. The earliest was the Jukes-Cantor model from the 1960s, which, as you said, did assume equal probability.
So it was a bit of a blunt instrument.
Exactly. It was a blunt instrument. But then the Kimura model was developed, and it introduced a vital biological reality: the difference between transitions and transversions, involving purines and pyrimidines.
Okay, let's pause and visualize this, because if you don't have a biochemistry degree, hearing purines and pyrimidines just sounds like alphabet soup.
It really does.
I like to think of the four letters of RNA, A, C, G, and U like biological Lego blocks. Adenine and guanine are your purines. They are large, bulky, double-ringed blocks. Cytosine and uracil are your pyrimidines. They are much smaller, single-ringed blocks.
That's a perfect way to look at it. So a transition mutation is when nature accidentally swaps a big block for another big block, or a small block for another small block.
Because they're similar.
Right. Because they are structurally similar in size, this substitution happens relatively easily without destroying the overall architecture of the RNA strand. It fits the wall you are building.
Makes sense.
But a transversion is when nature tries to swap a big block for a small block. This physically distorts the entire spiral staircase of the RNA. It causes a bulge or a pinch. It requires way more biochemical energy to force that fit, so it is biologically much rarer.
Right. So the Kimura model adjusts the math to tell the computer algorithm, hey, assume that transitions happen much more often than transversions. It weighs the branches of the family tree differently.
Yes. And when researchers are looking at the proteins these genes create, the sources mention they use reality check matrices, specifically something called the Bolosum 62 matrix.
I was reading about Blossom 62 and getting completely lost. Why does the algorithm care so much about this matrix?
Because it's an evolutionary reality check. If a mutation radically changes a protein's 3D shape, say by swapping a water-repelling amino acid for a water-attracting one, the protein will likely fold incorrectly, it will break, the virus will die, and that mutation will never be passed on to the next generation.
Oh, so it just gets weeded out immediately?
Exactly. Bolosum 62 assigns high positive scores to conservative, chemically safe substitutions and steep negative scores to radical, structurally dangerous ones. It ensures the computer doesn't accidentally build a family tree based on hypothetical mutations that would have biologically destroyed the virus in the real world.
Okay, that makes total sense. It keeps the computer grounded in biological physics. And when you apply all these incredibly sophisticated tools, the Kimura models, the Bolosum 62 matrices, the maximum likelihood algorithms to the raw genetic code of SARS-CoV-2, you get two competing frameworks for its origin.
Right.
Let's look at the evidence for natural spillover first. The texts show this framework relies heavily on a phenomenon called recombination.
Right. Recombination being nature's ultimate copy and paste mechanism. When two different strains of a virus happen to infect the same cell in the same animal at the exact same time, their genetic material physically tangles together.
So they get crossed.
Yeah. The replication machinery gets confused and jumps tracks midway through, copying creating a chimeric hybrid virus.
And the phylogenetic trees built by these algorithms show that the overall viral backbone, the chassis of SARS-CoV-2, closely matches bat coronaviruses like RATG13 and the banal 52 strain found in caves in Laos. However, The critical receptor binding domain, that highly specific grappling hook with the bulky rings and salt bridges we just discussed, is 97% identical to coronaviruses found in smuggled Malayan pangolins, specifically the GD and GX strains.
So the natural emergence hypothesis argues that an intermediate host animal was co-infected by a bat virus and a pangolin virus, and nature, through a random recombination event, spliced the highly optimized pangolin grappling hook onto the rugged bat chassis.
It makes perfect biological sense. We see recombination in nature all the time. But I have to push back on the missing links in this theory, because looking at the geographic data, there is a glaring anomaly that the sources highlight.
There is.
The closest bat relatives were found deep in southern regions like Yunnan province in Laos, which is over 600 miles away from the initial outbreak in Wuhan. And the pangolins were intercepted in completely different regions entirely.
That is a significant geographic hurdle.
It's massive. I mean, for natural recombination to occur, these geographically isolated animal populations would have to cross paths, swap respiratory droplets simultaneously, and then that perfectly assembled chimeric virus would have to somehow travel 600 miles to a dense urban center without leaving a single trace of an outbreak in the rural villages along the way.
And you are identifying the exact limitation of phylogenetic trees.
Yeah.
They only show evolutionary distance in a vacuum. They do not show geographic distance.
Right.
The natural framework absolutely relies on an undiscovered intermediate host network, likely wildlife farms or illicit transport routes. And despite testing tens of thousands of animals in the wet markets and surrounding farms, that direct intermediate host carrying the fully assembled virus has never been found.
Which brings us to the data supporting the laboratory manipulation framework.
Right. And this argument doesn't just rely on analyzing the genome in a vacuum. It relies heavily on a highly specific, declassified 2018 DARPA grant proposal called Project Diffuse.
Yes, diffuse.
This was submitted by the EcoHealth Alliance in partnership with researchers at the Wuhan Institute of Virology.
The diffuse proposal is central to the lab leak analysis because it outlines a meticulously detailed multi-year blueprint. It explicitly proposed a 3-step pipeline for coronavirus research.
Let's walk through those steps.
Step 1. Venture into remote bat caves in southern China to sequence novel, high-risk coronaviruses. Step 2. Use advanced reverse genetics platforms to splice the spike proteins of those newly discovered bat viruses onto the genetic backbones of well-characterized viruses, literally proposing building chimeric bat backbones in a lab setting.
And step 3 is what makes the hair on the back of your neck stand up, because it addresses the most unique feature of SARS-CoV-2. The proposal explicitly requested DARPA funding to synthetically insert human-specific cleavage sites into these newly created chimeric bat viruses, specifically to test if it enhanced their ability to infect human airway cells.
So proponents of the directed laboratory evolution framework argue that SARS-CoV-2 looks exactly like the product of the diffuse pipeline. It has a bat-like genomic backbone, a swap receptor binding domain, and an unprecedented, perfectly tailored cleavage site that no other known virus in its subgenus possesses.
And again, I'm going to push back here because I think it's important to be rigorous.
Please do.
If the natural theory is missing the geographic intermediate host, what is the lab theory missing? The sources indicate a massive gap here, too.
It is missing the ancestral backbone.
Exactly.
To definitively, scientifically prove this virus was engineered, you must identify the exact starting material. You need to find a viral genome in a pre-2019 laboratory database that is 99.9% identical to SARS-CoV-2, simply lacking the cleavage site insertion.
Yeah.
You need the direct parental chassis.
And despite intense global scrutiny, hacking, and leaks, no such ancestral backbone has been definitively linked to any laboratory inventory prior to the outbreak.
So we are left in this incredibly frustrating position. Both frameworks, natural stochastic evolution and directed laboratory evolution, have massive, unexplainable missing links. We are looking at a biological machine with unprecedented capabilities, but the exact forge it was created in remains a mystery.
It does. But while the origin is debated, the mechanism of action is undeniable. And if the virus's entry mechanism into the cell is controversial, its method of hiding from the human immune system once it gets inside is truly chilling.
And this requires us to take a deep dive into the genetic alphabet and the terrifying concept of structural camouflage.
Right, and this brings us to the second major phase of our exploration today.
If we are talking about camouflage, we have to talk about the furin cleavage site, or FCS. We just mentioned that the diffuse proposal wanted to insert one, but what exactly does an FCS do?
Good question. Structurally, it is a tiny sequence of specific amino acids, guanine, arginine, alanine, or PRRR. The texts describe it as a pre-activation switch.
Okay, a pre-activation switch.
Think of the spike protein like a spring-loaded trap. For the virus to actually fuse with your cell membrane and dump its genetic payload, the top of the spike protein has to be physically severed to release the spring.
So it needs to be cut.
Yes. The PRRR sequence is a custom-designed molecular cutting board that perfectly attracts an enzyme called furin. Now, furin is completely ubiquitous in the human respiratory tract. Your lungs are full of it.
Right.
As the virus replicates, the host's own furin enzymes prec the virus, clipping the spike protein so it is spring-loaded and primed to infect the next cell instantly.
But the genetic fingerprint of this specific cleavage site is what has caused an absolute uproar in the bioinformatics community, and this requires us to look closely at the codon alphabet.
Yes, the codons are key here.
As we know, RNA is read by the ribosome in triplets called codons. 3 chemical letters equal 1 amino acid. The amino acid arginine, which is the crucial R in the PRRR sequence, can be spelled out using 6 different codon combinations. It's like having 6 different synonyms for the exact same word.
Exactly. And in the SARS-CoV-2 furin cleavage site, the virus uses the codon sequence CGG to produce arginine. In fact, it uses 2 of them back-to-back, a tandem CGG sequence.
Which is really unusual.
It is. The reason this raises a massive red flag in the evolutionary literature is that coronaviruses generally exhibit a powerful evolutionary bias against using cytosine guanine-ending codons. They heavily avoid the sequence CGG.
And they avoid it because of ZAP, right, the zinc finger antiviral protein.
Yes, ZAP.
The literature describes ZAP like a molecular heat-seeking missile in the mammalian immune system that specifically hunts down and shreds RNA strands containing CGG sequences.
Yeah.
So over millions of years, bat coronaviruses evolved to drop those specific letters from their vocabulary to fly under the radar.
That is the established evolutionary pressure.
Yeah.
If you use CGG, ZAP finds you and destroys you. Yet the FCS in SARS-CoV-2 contains this highly unnatural tandem CGG-CG sequence.
Okay.
And what makes it so striking is that while bat coronaviruses hate CGG, human cells absolutely love it. The CGG codon is the exact sequence humans use most frequently. It accounts for roughly 21% of all human arginines. Human molecular machinery is highly optimized to read and translate it quickly.
So you have a virus that normally avoids this sequence like the plague, suddenly sporting a tandem sequence optimized perfectly for human translation, right at the most critical junction of its spike protein.
It's a glaring anomaly.
But the CGG anomaly, as weird as it is just one piece of the disguise. Because the true horror of this pathogen's evasion strategy is molecular mimicry.
To understand molecular mimicry, we first have to understand how the human body tells friend from foe. We have to look at the human thymus.
I love the analogy the sources use for this. Think of the thymus, which is a small organ located right behind your sternum, as an extreme, unforgiving military boot camp for your immune system's T cells.
It's a brutal boot camp.
When your body creates a naive T cell in the bone marrow, its entire job is to patrol the bloodstream and attack anything foreign. But before it is ever released into the general circulation, it must pass through the thymus boot camp.
Right.
Inside the thymus, the body presents the young T cell with a vast, comprehensive catalog of every single protein your body naturally makes. Heart proteins, brain proteins, liver proteins. It's basically the drill instructor holding up flashcards saying, here's what our uniform looks like, memorize this.
And the stakes of this boot camp are absolute. If a young T cell reacts to any of those self-proteins, if it attacks the catalog, it fails the test and is immediately destroyed by the body.
Wow.
This brutal microscopic selection process is called clonal deletion. It ensures the immune system has a built-in systemic blind spot for its own tissues, which prevents widespread autoimmunity.
So molecular mimicries is the virus intentionally hiding inside that blind spot?
Yes, it is wearing the uniform. But the theoretical models in the sources offer 2 distinct hypotheses for why this evolves in nature.
Okay, what are they?
The costly autoimmunity hypothesis suggests the mimicry is explicitly for evasion. The virus wears the biological uniform of human tissue, slipping past the initial immune dragnet because the guards have been ordered not to attack it.
But there's a cost.
Right. The costly part happens when the disguise eventually fails. When the virus replicates so much that the immune system finally realizes it's under attack, it goes into overdrive and produces massive amounts of antibodies against the virus. Because the virus looks exactly like human tissue, those newly minted antibodies cross-react and attack the host's own body, leading to devastating autoimmune disease.
The cure becomes the disease.
Exactly.
And the alternative is the mimicry trade-off hypothesis, which says the immune evasion is just a happy, horrifying accident. The virus just structurally needs to hold a specific 3D shape to bind to our cells, and that shape just happens to match a human protein. It's trapped in that shape for functionality.
Right, it's not intentional, it's just physics.
Regardless of the why, the what is terrifying. The analyses in our SAC focus heavily on a specific 5 amino acid sequence located on the spike protein called the RLFRK sequence: arginine, leucine, phenylalanine, arginine, lysine.
And what is truly chilling about the RLFRK sequence is what it mimics. The bioinformatics show it is a perfect structural mimic of 2 human proteins, POMGNT1 and TENM4 or TENM4.
POMGNT1 and TENM4.
Now, these aren't just random proteins. These are vital regulators of myelin in the central nervous system.
Right.
Myelin is the protective fatty insulation wrapped around your nerve fibers, allowing electrical signals to travel quickly. POMGNT1 and TENM4 are not just structural bricks in the wall, they're the active regulatory machinery, the cellular foremen who manage the construction and maintenance of your entire nervous system.
This is a light bulb moment.
Yeah.
So when the human immune system inevitably develops antibodies against the viral RLFRK sequence, those antibodies circulate, eventually cross the blood-brain barrier, and begin assassinating the cellular foreman, maintaining your nerve insulation.
That's exactly what happens.
It perfectly explains the profound, lingering neurological symptoms of long COVID. The severe brain fog, the loss of smell, the peripheral neuropathies. The virus primes your immune system to literally hunt your own brain.
It does. And the mimicry isn't limited to just the RLFRK sequence. The sources highlight another viral component called ORF8.
ORF8.
Unlike the structural proteins stuck to the virus, ORF8 is heavily secreted. The virus actively pumps it out into the host's bloodstream. It acts as a viral mimic of a human cytokine called interleukin-17A, or IL-17A.
And cytokines are the chemical messengers of the immune system. IL-17A is our cellular alarm system. Its job is to recruit massive amounts of immune cells to a site of severe infection.
Exactly. By actively secreting a mimic of IL-17A, the virus literally presses the body's own panic button. It triggers a massive, unregulated inflammatory response, leading to what we know as a cytokine storm.
Oh, cytokine storm. We heard a lot about that.
The immune system overreacts so violently to the fake signals that it floods the lungs with fluid and immune cells, causing massive tissue damage. The virus tricks the host into suffocating itself.
But here's where we see evolution occurring in real time, right before our eyes. The epidemiological sources document that as the virus circulated in the human population, it underwent reverse evolution.
Yes.
Researchers observed variants popping up in Singapore with a massive chunk of missing code in the ORF8 gene, known as the Delta 382 deletion. And patients infected with this broken panic button actually experienced significantly milder infections.
It perfectly illustrates the reverse evolution framework. The original ORF8 was causing too much inflammation. It was killing the host too quickly or putting them in an ICU, which physically isolates them and stops the virus from spreading.
So it's bad for the virus to be that deadly.
Right. So the evolutionary pressure forced the virus to aggressively shed that trait. It deleted the ORF8 sequence to become milder, allowing the host to walk around coughing and transmitting the virus further.
Yeah.
The virus actively scales back its own weaponry to ensure its long-term survival in the population.
It's an incredible adaptation. Now, I want to pivot here because while we've been talking about the wild virus, there is a massive, highly technical distinction regarding the RLFRK sequence that we just discussed.
This is a vital distinction to make.
The bioinformatics data reveals a crucial intersection between the wild virus and the mRNA vaccines that rolled out in 2021.
In the natural, wild SARS-CoV-2 genome, the virus codes for the RLFRK sequence using standard bat coronavirus codons. Specifically, it uses the codons AGA and CGA to spell out the arginines in that mimicry sequence.
Right, the normal viral spelling. No weird CGG anomalies there.
Exactly. However, when Moderna and Pfizer engineered their mRNA vaccines, their primary goal was to maximize the production of the spike protein in human cells to generate strong, rapid immunity.
That makes sense.
To achieve this, they utilized a standard biotechnology process called codon optimization. Their bioinformatic algorithms systematically swapped out the virus's preferred slower codons for codons that human ribosomes translate much, much faster.
And because human cells overwhelmingly prefer the CGG codon for arginine, as we established with the furin cleavage site, the manufacturing algorithms automatically swapped the wild viral code for CGG codons at the RLFRK site in the vaccine mRNA.
Precisely. And the implication is profound at a molecular level. By optimizing the codons to CGG, the vaccine RNA now perfectly genetically mimics the human POMGNT1 and TENM4 sequence, not just at the amino acid level, but at the exact nucleotide sequence level.
So it's an identical match.
Yes.
Yeah.
Combined with something called prefusion stabilization. These are the 2P mutations that lock the vaccine spike protein in a rigid, highly visible shape so the immune system can see it. This optimization drives massive translation speed and protein longevity.
So the vaccine creates a much higher density of these specific proteins in a shorter amount of time than a natural, slowly replicating wild viral RNA strand would. And according to the immunology models in the stack, this massive kinetic burst of translation increases the quantitative risk of rare, cross-reactive autoimmunity in susceptible individuals. To be clear, the models showed the wild virus ultimately inflicts vastly more systemic damage because it actively replicates, destroys lung tissue, and triggers cytokine storms.
Right. The wild virus is much worse overall.
But the optimized mRNA creates a unique, hyper-efficient exposure to that specific neurological mimicry sequence.
Which perfectly transitions into the final variable in this complex molecular equation. Human genetic vulnerability.
Yeah.
We all lived through this, and the constant question was, why does one person experience a mild 2-day cold while another experiences a catastrophic mimicry-induced autoimmune collapse? The virus is a constant key, but the exact shape of the human lock varies drastically.
The sources look closely at missense variants in the ACE2 receptor. A missense variant is basically a single-letter typo in our DNA that subtly changes the 3D shape of our cellular doorways.
Right.
The data shows that a variant known as G2 of an R, which is found more frequently in European populations, changes the receptor shape to increase the virus's binding affinity by a massive 7.6 kilocalories per mole.
And in the realm of physical chemistry, a 7.6 kilocalmol shift is astronomical. It represents an almost inescapable magnetic attraction.
So they're just far more vulnerable.
Yes. For these specific individuals, the virus requires vastly fewer particles to initiate a severe, deep-seated infection. Their doors are essentially wide open. Conversely, the K26R variant, found more frequently in Ashkenazi populations, alters the shape to slightly decrease the binding affinity, providing a subtle layer of natural resistance.
But reading this, I kept wondering, how do epidemiologists figure out who has these lethal vulnerabilities? Because you run into a massive statistical hurdle called survivorship bias.
Survivorship bias is a huge problem.
All right. Right. If a novel pathogen causes rapid, severe mortality in people with a hidden genetic vulnerability, an ablation architecture that permanently removes them from the population, studying the survivors gives you completely skewed data. You are studying the armor, not the chink in the armor.
Exactly.
Because the most vulnerable victims are gone, you can't exactly ask them to participate in a DNA sequencing study.
The classic epidemiological trap. To solve this, researchers use highly advanced statistical tools. They use family-based association studies and sibling proxies.
Sibling proxies.
Because you share roughly 50% of your DNA with a full sibling, researchers can sequence the genomes of a deceased victim's surviving siblings and parents. By analyzing the inherited blocks of DNA, the haplotype statisticians can computationally reconstruct the missing genetic hardware of the deceased. They basically build a highly accurate virtual genomic profile of the victim.
They construct the ghost in the machine. That is brilliant.
It really is.
And they pair this with a technique called Mendelian randomization. Because your genes are assigned randomly at conception, they are immune to environmental confounders like smoking, diet, or socioeconomic status.
Right. It filters out lifestyle choices.
By tracking these genetic variables, scientists can prove a direct, undeniable causal link between a hidden genetic trait and severe disease outcomes, completely bypassing the noise of lifestyle choices.
So having established the hyper-precise, terrifyingly complex reality of the virus, the sabotage, the mimicry, the binding affinities, and the subtle variations in human genetics, we must now shift our focus to the macro level.
Yes, the macro level.
If this virus is so incredibly camouflaged that it tricks the human body, how on earth were our government safety databases supposed to spot the side effects when the vaccines rolled out?
Exactly. This is where we transition from biology to bureaucracy. We have to look at how the United States government attempted to measure the safety of the massive vaccine rollout designed to stop this pathogen.
And the primary safety net is the V8URS database.
The Vaccine Adverse Event Reporting System. The texts describe it as a passive surveillance net, meaning it just sits there on a server waiting for doctors, nurses, or patients to actively submit reports.
It's incredibly noisy.
Right. People report everything from sore arms to car accidents. To find the true causal safety signals in that ocean of noise, the FDA relied on a mathematical algorithm they considered their absolute gold standard, the MGPS, or multi-item gamma Poisson shrinker.
The MGPS is an incredibly complex piece of math that relies on Bayesian statistics. Its entire function is to constantly compare the observed number of adverse events for a specific vaccine against a baseline of expected events drawn from the entire historical database.
Okay.
It uses a Poisson distribution to model the probability of rare events occurring and a shrinker function. The shrinker is basically a mathematical anchor. It pulls extreme, alarming values from small sample sizes back toward the baseline, preventing the FDA from constantly reacting to false alarms.
It's designed to be a very cautious, deliberate, conservative filter. You don't want to pull a vaccine off the market because of a statistical fluke.
Exactly.
But the Senate report outlines a fatal mathematical flaw in this gold standard when it collided with the reality of 2021. And I want to use the hemlock versus arsenic analogy here because reading the mathematical formulas made my eyes cross, but this analogy beautifully illustrates the statistical concept of masking.
Yes, let's walk through the masking effect carefully, because it is the linchpin of the entire governmental failure.
Imagine you are a toxicologist tasked with testing the toxicity of a rare new poison, hemlock. Normally, you'd compare hemlock to a harmless baseline like saline solution.
Right.
If hemlock causes rapid cellular death and saline doesn't, the math screams at you. You have a clear, undeniable safety signal. But what if your baseline is suddenly polluted?
What if it's not harmless saline anymore?
Exactly. What if, instead of harmless saline, your baseline is heavily flooded with data from arsenic poisoning? If you test hemlock against a baseline that is artificially inflated by arsenic, the hemlock no longer looks like a statistical anomaly. Relative to the new, highly toxic baseline, the hemlock blends right in. It looks normal.
And that analogy maps perfectly onto the FDA's data parameters in 2021. In a normal year, VARs receives reports for flu shots, tetanus shots, things with very low adverse event profiles.
Right.
But in 2021, an overwhelming 99% of all reports submitted to VARs were for COVID-19 vaccines. Now, the FDA had established a strict mathematical threshold for a safety signal. The EBO5 score had to exceed 2.0.
Meaning the adverse event had to occur at more than double the expected baseline rate to trigger a warning.
Yes, but if you are testing the Moderna vaccine for a severe adverse event, say myocarditis, the algorithm compares it to a baseline overwhelmingly comprised of reports from the Pfizer and Johnson & Johnson vaccines.
Oh, I see.
If all these new vaccines are causing a similar elevated rate of that specific side effect, the baseline is massively inflated. Reaching that EBO5 threshold of 2.0 became a mathematical impossibility. The algorithm was effectively deactivated by the sheer volume of shared side effects. The signals canceled each other out.
The algorithm was completely blinded. The anomaly became the baseline.
It did.
But the true tragedy detailed in the subpoenaed sources is that the FDA had the exact experts and the exact tools needed to fix this blind spot internally. The report introduces the internal whistleblowers. Dr. Anna Scharfman, a highly decorated senior medical officer and top data mining expert at the FDA, and Dr. William DeMuschel, who is the literal inventor of the original MGPS algorithm.
These aren't outside critics screaming on social media. These are the architects of the machine inside the building.
Exactly, and they didn't just point out the flaw, they brought the solution. They introduced a superior updated model called RGPS, the regression-adjusted gamma Poisson shrinker.
Regression adjustment is a powerful statistical technique that isolates variables. Think of it like a mixing board in an audio recording studio.
I love that analogy.
The old MGPS system just looked at the master volume output. If the master track was loud, it couldn't tell if the drums, the guitars, or the vocals were causing the spike. But RGPS mathematically isolates the individual tracks.
Okay, so it separates them.
It models the effects of multiple covariates simultaneously, like age, gender, and the presence of other similar vaccines in the database, and builds a clean, hypothetical baseline to test the data against. It strips away the distortion.
It puts on noise-canceling headphones to tune out the arsenic so you can clearly hear the hemlock. And the findings were staggering. By applying RGPS to the raw data, Dr. Scharfman and Dr. DeMuschel uncovered 49 distinct examples of extreme masking. By stripping the distortion, the new math illuminated 25 hidden, statistically significant safety signals that the old system was entirely blind to. And these were not mild signals like a sore arm. They found sudden cardiac death, acute myocardial infarction, pulmonary infarction, and Bell's palsy.
Which brings us to the most uncomfortable part of this deep dive, because data on a spreadsheet doesn't exist in a vacuum. Human beings have to look at that data and act on it.
Right.
What happened when the architects of the system brought a flashing red warning of sudden cardiac death directly to the FDA leadership?
The timeline of the internal blockade in 2021 is incredibly damning. Let's pause and really look at these dates. On March 1st, Dr. Sharfman officially briefs the CBER director, Dr. Peter Marks, presenting a slide deck proving the old method was broken. On March 26th, she sends the top officials the actual spreadsheet detailing the 49 examples of extreme masking and the hidden cardiac signals. She has moved from theoretical math to terrifying clinical reality. If I sent that email, I'd be waiting by the phone for the emergency alarm to sound.
But the institutional response was evasion. By early May, the officials weren't investigating the pulmonary embolisms. They were figuring out how to manage Dr. Sharfman.
Manage her.
Yes. On May 7th, she received a formal cease and desist email from top CBER officials, including Dr. David Menick. They explicitly directed her to hold off on creating and sending data mining reports. They ordered their top data expert to stop looking at the safety data.
A cease and desist? Are you kidding me? From her own agency?
Yeah, from her own boss.
But she didn't stop. On July 12th, she alerts them that the RGPS method has detected an increased mortality signal death and sudden death. You would think that triggers a 5-alarm fire drill. Instead, on September 15th, Dr. Peter Marks escalates his administrative complaints, telling her boss that she needs to cease and desist because her persistent data reports have become a major distraction.
The motivation behind this wall of silence reveals a dark institutional reality that we often ignore in public health. First, there was a structural turf war. Dr. Sharfman worked for CDR, the Center for Drug Evaluation and Research, while vaccine oversight belonged to CBE, the Center for Biologics Evaluation and Research.
So they were just protecting their turf.
CBER officials constantly complained she was out of her lane, prioritizing their organizational hierarchy over her undisputed expertise. But the more pervasive, insidious fear was public relations.
Yes, in that September 15th email, Dr. Marks writes the most revealing sentence in the entire Senate report. He explicitly justifies shutting her down by stating her data could feed into anti-vaccination rhetoric.
That's the quote.
They were actively weighing the optics of the safety data against the reality of the safety data. It is a tragic inversion of public health. They chose to protect the PR narrative, fearing public panic and vaccine hesitancy, over unmasking the raw data that pointed to fatal side effects.
And privately, the texts show they knew she was mathematically correct. Dr. Menick, who helped draft the gag order, drafted an article for the medical journal The Lancet in late 2021, where he explicitly admitted that the FDA's data mining scores could be muted by the volume of COVID vaccine reports contributing to the comparator group.
He admitted it.
He did. But that admission was scrubbed from the draft before publication.
Furthermore,
as FOIA requests and congressional inquiries piled up in the summer of 2022, the FDA took a drastic step. In July 2022, they simply stopped sending the weekly data mining reports to the CDC altogether.
just stop sending them.
They severed the interagency data pipeline to evade external legal scrutiny.
This bureaucratic suppression sets the stage for the explosive confrontation detailed in the June 2026 letter from Senator Ron Johnson. The senator issued a massive subpoena to the Department of Health and Human Services, resulting in millions of pages of records. The subcommittee used sophisticated e-discovery software to hunt for anomalies, and they extracted internal FDA email threads from December 2025 regarding pediatric deaths.
And here we shift from the sterile mathematical world of data mining into the agonizing, tactile world of medical forensics. By late 2025, the FDA's Center for Biologics Evaluation and Research conducts a retrospective review of 10 pediatric deaths that were initially assessed as probably or possibly connected to the mRNA injections.
Right.
The December 5th email from Dr. Magna Elamchandani documents a painstaking, rigorous quality control review designed to reduce subjective variability and establish ironclad clinical consensus among the pathologists.
And they walked through the clinical pathology to show the extreme medical rigor. They weren't just rubber stamping these. Case 1 was downgraded to unassessable. The autopsy was incomplete, but the death certificate noted florid lymphocytic myocarditis, an intense immune swarm destroying the heart tissue.
But there's a confounder.
Right. The child also had an active wild SARS-CoV-2 infection. Because both the vaccine and the wild virus are known triggers for myocarditis, the massive confounding variable created an impenetrable diagnostic wall. It was a biological tie, so they threw it out.
Case 2 is also downgraded to unaccessible due to contradictory information between the death certificate and the autopsy report, as well as the presence of parvovirus, a completely different pathogen known to cause pediatric myocarditis.
So the evidentiary chain was broken, so it was discarded.
Exactly. Case 3 was downgraded to unlikely. A pediatric cardiologist reviewed the autopsy and recognized the likely cause of death as HOCM hypertrophic cardiomyopathy.
And HOCM is a structural genetic architectural thickening of the heart chamber walls, which is entirely different from the acute inflammatory immune swarm of myocarditis. So by strictly filtering out confounding viruses, contradictory records, and underlying genetic conditions, the rigorous review downgraded the 10 cases to 7.
But because the review process was so unbelievably strict, the 7 surviving cases represented a profound, undeniable safety signal that withstood the absolute strictest scientific scrutiny the agency could apply.
They were ironclad.
5 of those remaining pediatric deaths explicitly involved myocarditis. And based on this, Dr. Aline Chandani explicitly recommends taking massive regulatory action. She writes that their internal recommendation is to trigger a class SLC, a safety labeling change, to formally add myocarditis with fatal outcomes to the mRNA vaccine warning labels.
A warning label is a foundational legal contract. It shifts the entire liability landscape and instantly alters the standard of care, forcing pediatricians across the country to explicitly disclose the risk of fatal outcomes to parents before administering the shot.
It's a huge deal.
But how does a label warning actually get debated and approved inside the FDA? Dr. Alim Chandani noted they would present the recommendation at a December 11th Safety Working Group, or SWG, meeting.
The SWG meeting acts as a bureaucratic trial by fire. The Safety Division acts as the prosecution, presenting the hard forensic evidence of the pediatric deaths. But the room is also filled with public relations directors, economists, and policymakers acting as the defense.
Right.
They weigh the forensic clinical data against the macroeconomic and epidemiological PR fallout, asking a grim utilitarian question. If adding fatal outcomes to the label causes a drop in vaccine uptake, will that result in more viral deaths than the vaccine side effects?
And this brings us to the ultimate collision in the summer of 2026. 6 months after that December SWG meeting, the label change remains a complete black box. It is unknown if the fatal warning was ever formally approved. Yet while that debate is shrouded in secrecy, the CDC moves forward and awards $1.24 billion in new contracts to Pfizer to secure vaccine supply for the upcoming fiscal years.
Senator Johnson's June 16 letter is born out of this incredible friction. You have the rigid forward momentum of government procurement signing billion-dollar checks directly colliding with the ignored, flashing emergency brakes of the FDA's Internal Safety Division.
It's a massive collision.
He demands the unredacted communications in the SWG meeting minutes to find out exactly how a clinical recommendation for a fatal outcomes warning was swallowed by the bureaucracy.
So to summarize the incredible journey we've taken through these sources today, we started by looking at the microscopic physics of the virus, how the bulky phenylene ring and the glutamine salt bridge in the S1 domain mathematically lock onto our cells. We explored how the virus hides from our immune system using the RLFRK sequence to maliciously mimic our own myelin regulators, POMGNT1 and TENM4, and how that structural reality collided with our own genetic variations. We then transitioned from the biological machine to the bureaucratic machine, detailing how the massive flood of pandemic data fundamentally broke the MGPS algorithm through the masking effect. And finally, we traced the chilling paper trail of internal FDA emails, watching as leadership actively suppressed the state-of-the-art RGPS fix, gagged their own top data experts to protect a PR narrative, and ultimately ignored the forensic reality of fatal pediatric myocarditis while signing billion-dollar procurement contracts.
It is an overwhelming amount of information to synthesize. But I want to leave you with a chilling societal extrapolation that builds on the fundamental flaw we uncovered today.
Oh, absolutely.
We spent the last hour looking at how a massive volume of data can completely hide the truth by artificially inflating the baseline, effectively blinding the absolute smartest experts in the room. In our modern age, we increasingly rely on artificial intelligence, machine learning, and opaque algorithms to filter our economy, to monitor our infrastructure, and to curate our daily newsfeed.
That's true.
These systems are all built on mathematical baselines. What happens when the anomaly itself slowly, quietly becomes the new baseline because of a flood of distorted data? Who is checking the math on the machines that run our world, and what other critical, life-altering warnings are currently blending perfectly into the everyday noise?
That is a deeply unsettling question to ponder. When the X-ray machine is broken and the waters are murky, the microscopic magic tricks don't stop, they just go unseen. Thank you for joining us on this deep dive. Keep reading, keep questioning the consensus, and never stop asking the hard questions about the data behind the headlines. Until next time.